purpose of this video is to give a quick introduction to the Plant Metabolomics Resource website, as well as give a brief example of how to access some of the data that is available. Upon reaching the website, what you'll see is several boxes. One of these discussing what the resource is about, a box describing news, such as new species or data that is available, another box describing how to cite the resource when used in your own work, and down here on the right, a brief overview of what is metabolomics, as well as a text description of how to use the resource, and a link to an annotated explanation of the type of figures that are used to present data for comparisons between experiments. And then on the left here, we see the various species that are currently available, and if we click on one of these, we can see the particular experiment that the data has become available from. In this case, we've selected Echinacea purpurea. We have a link to a species overview, and then we also see the experiments from which data is available, in this case from the Additional Plant Consortium. On the left, we have the various samples or tissue types that our data is available from, the number of replicates for each sample. On the right here, we have an area for metabolite search, where we can search by metabolite name if it's been chemically defined in the experiment. We also have a range search function, which allows you to search a range of certain mass over charges or particular retention time. We scroll down. On the right, we see an area where we can generate a volcano plot, as well as a link to a description of the volcano plot. The volcano plot allows comparison of metabolites between samples. Further down, we see the biological material, such as the growth conditions, variables tested, and harvest info. And on the left, we see the various platforms from which the data has been generated. To give an example of how to search for a particular metabolite, we'll go back up to the search bar. In this case, see if the amino acid serine has been chemically defined in this experiment, and it has. Alternatively, we could have also searched for using the mass over charge ratio of serine, and we'll bracket the exact mass of serine, which is 105, with 104 to 106. And again, serine is returned. If we click on the particular metabolite, we'll see several tabs that appear that can provide basic compound information. We can conduct a chem spider search, but most importantly, we'll see experimental data on the species and experiments that are available. So in this case, we see the metabolite, the particular platform used, the species, the experiment, and a histogram showing the relative abundance of the particular metabolite across various replicates and across various biological samples or tissue types. If we click Go to Omics Analysis, this takes us to our correlative analysis of metabolomics and transcriptomics data for the species and the platform. We can also change the metabolite here, change the platform for the omics analysis, as well as various settings, alter the p-value for the correlative analysis, or r-value and false discovery rate. And then down below we see the various transcripts which have the highest correlation to metabolite levels in each biological tissue sample. An annotation for that transcript if it's available, the Pearson correlation coefficient, and a p-value. We'll just click the second sequence ID here, and what we see is the correlation using a log to abundance across the various tissue types of the metabolite and the particular transcript we've selected. If we scroll down, we can see a predicted amino acid sequence for this transcript, as well as the nucleotide sequence for the transcript, and the annotation of information if it's available. I hope this brief video has been informative and helpful, and if you have any problems using the Plant Metabolomics resource, please contact Eve Wortel, and her email address is available at the bottom right of the news section on the front page. Thank you.